Welcome to amino acid catabolism. Now, amino acids are going to be broken down in a couple of different ways, basically to just break down and reuse nitrogen. Nitrogen in the form of ammonium, which is NH4. NH4 plus, mind you. There we go, that looks a little better. All right, so amino acid catabolism is going to happen in a couple different states when you have a high turnover, so the proteins that are in your body is being broken down and rebuilt, where you have a lot of a lot of amino acids floating around. So they're going to break down into these amino acids. They're going to be in your system. They're going to be used to remake some protein. These amino acids that are in your system are there. They can be broken down. Another case is in a protein-rich diet. Obviously, you intake a lot of protein, you're going to make more amino acids. Those amino acids that are not used for creating new functional proteins are going to be digested. We do not have a way to store energy in the form of amino acids because we use amino acids for the proteins that basically allow us to structurally and functionally exist as human beings, or just basically animals. The third state is in a case of starvation or uncontrolled diabetes. Starvation, you have no sugars, no fats, you're going to start breaking down whatever your body's got available. When you run out of glycogen, when you run out of fat, you're going to turn to all the proteins in your body. So in starvation or diabetes mellitus, in the case of diabetes, your body just doesn't recognize that it's getting sugar. So if it doesn't have any intake to the cell, you're going to have a high blood sugar anyways, but your body doesn't recognize that because it can't get any kind of sugar into the cell, so it starts breaking down your proteins to try to get some kind of energy into your cells. So really quickly, what that kind of looks like is we've got a lot of dietary protein coming in. So we've got, let's just go right here, we've got intracellular proteins. Now these intracellular proteins are going to be the ones in your cytoskeleton, all of your enzymes, that kind of thing. They can be broken down into amino acids. Your dietary protein can be broken down into amino acids. And these amino acids are going to go to a couple of places. They can be broken down further into the carbon skeleton, which is basically going to be used for energy. And it can also be broken down into when you take off that carbon skeleton, all you're really left with is that alpha amino group in the form of NH4. That can be used for the synthesis of other amino acids aside from the one that was broken down and their eventual return into intracellular proteins, or it can be excreted via the urea cycle. The urea cycle will be a separate video. What we're going to talk about right now is basically this NH4, how it comes off of the amino acid and where it goes. Spoiler alert, it's broken down in extrahepatic tissues and goes to the liver, specifically to the mitochondria. So, let's talk just a little bit about extrahepatic tissues. Extrahepatic tissues. In extrahepatic tissues, you're going to be producing NH4 plus or ammonium. Why we're producing ammonium isn't particularly stated. Ammonium is toxic. It can form a nitrate or nitrite, which can have some DNA mutagenic effects. I wrote down a couple other things, but that's the one that's like kind of most prevalent. DNA mutation is never a good thing. So this NH4 is toxic and it needs to be brought somewhere to be broken down. In our bloodstream, we have a lot of glutamate. We have a high concentration of glutamate in our bloodstream. So what's going to happen is this NH4 in tissues is going to be bound to glutamate. So if we've got glutamate here, it's going to be changed into something via the addition of this ammonium group. Glutamate is an amino acid that looks a little bit like this. And it's got basically a double bonded oxygen and an O-. Glutamate is going to be changed into glutamine via the enzyme, you guessed it, glutamine synthetase. So this is glutamine synthetase. Now if we remember in class that synthetase means that we are using ATP, in this case we are going to use all of 1 ATP. Why are we using ATP? The hydrolysis of one of those high energy phosphate bonds is going to allow us to bind this nitrogen to the glutamate, forming glutamine, which is going to look a little something like this. You basically take this hydroxyl group and tack on a nitrogen instead, and you've got glutamine. Obviously the rest of the amino acid is over there, not super worried about it for right now. What's going to happen with this glutamine, it's going to be formed in extrahepatic tissues. What's going to happen then is it's going to get pumped into the bloodstream. It's going to be pumped into the bloodstream and it's going to be carried places. Where is it going to be carried? Give me a half second and we're going to find out. So, that glutamine that is in the bloodstream can go to 3 main locations. It can go to the intestines, to the kidneys, and to the liver. The only thing we really talked about in class was the liver itself, so that's where we're going to spend the vast majority of our time. That glutamine, once it reaches the liver, is going to undergo a chemical reaction. What's going to happen is you're going to pump out that nitrogen to form glutamate again. This is nitrogen transport, bringing nitrogen that's produced in extrahepatic tissues back to the liver for processing. This enzyme, we are taking glutamine and we are breaking it apart into nitrogen and glutamate. The enzyme here is actually glutaminase. If we think about phosphatase, it's going to be breaking off a phosphate group, therefore glutaminase is breaking apart glutamine. Now, that was really fun. So this NH4 is going to be channeled directly into the urea cycle, or it can be used to synthesize new proteins, or new amino acids, mind you. Probably the urea cycle for processing, but you never know. The liver does pretty much everything. And glutamate is going to be dehydrogenated further. Via the actions of glutamate dehydrogenase, we are going to split apart. I think I'm wrong on that.